are paper slide videos on diabetes and insulin resistance, created by Koczwara, Reiner, and Sandra Saji. What is diabetes? Well, diabetes is a disease when a person's blood glucose sugar is too high, or in other terms, when the body doesn't react positively towards insulin. This can result in an increase in an abnormal amount of carbohydrates, glucose, and urine. Insulin, a hormone made by the pancreas, helps glucose from food get into your cells to be used for energy. Sometimes your body doesn't make enough or any insulin, or doesn't use insulin well. Glucose then stays in your blood and doesn't reach your cells. Over time, having too much glucose in your blood can cause health problems. The diagram shows the basic idea of how diabetes are formed. Specifically, at number 1, the stomach and small intestine break down carbohydrates and food to glucose. At number 2, glucose enters the bloodstream. At number 3, gastric hormones promote insulin secretion in response to glucose. At number 4, the pancreas produces insulin, but in type 2 diabetes, the body is resistant to its effects. At number 5, glucose is unable to enter the body cells effectively. And finally, at number 6, glucose builds up in the bloodstream, damaging blood vessels. Although diabetes has no cure, you can take steps to manage your diabetes and stay healthy. Careful meal, daily exercise, and taking diabetes insulin to control blood sugar levels. Insulin is very important. In skeletal muscles and liver cells, glucose is stored as glucagon. Insulin stops the use of fat as an energy source by preventing the release of glucagon. In a normal body, cells constantly send out and receive signals, which the signals may come from the environment, but they can also derive from other cells. For a response to happen, the cells have to be triggered, which means the signals have to be transmitted across the cell membrane. Specifically, the signal works by reacting with receptor proteins in both the inside and outside of the cell. In type 1 diabetes, the pancreatic cells that produce the insulin are lost. Therefore, the insulin signal is also lost, which leads to the accumulation of sugar at toxic levels in the blood. Both type 1 and type 2 diabetes are similar in symptoms, but they are caused in different ways. Type 2 is a milder form of diabetes 1. In type 2 diabetes, the body has lost the ability to respond to the insulin, which leads to the result of the blood sugar levels becoming dangerously high. This is long-distance signaling because hormones usually travel the circulatory system to target cells, which does not occur in diabetes. Several hyperglycemia-induced mechanisms may induce vascular dysfunctions, including increased formation of 2 fatty acid chains bonded to a glycerol molecule through ester linkages, and the subsequent activation of protein kinase C isoforms and accelerated non-enzymatic formation of advanced glycated end products. It is likely that each of these mechanisms may contribute to the known features of diabetic complications. Diabetes alters the C-AMP protein kinase cascade system. Mitogen-activated protein kinase cascade is activated in mesangial cells cultured under high glucose conditions. The activation of protein kinase C found in these cells has been proposed to contribute to the development of diabetic nephropathy. However, the abnormalities distal to PKC have not been fully clarified yet. Mitogen-activated protein kinase cascade, an important kinase cascade downstream to PKC, and an activator of cytosolic phospholipase A2 by direct phosphorylation. Cell-to-cell communication can also be shown through the blood levels throughout the body. For example, in a normal human body, when blood sugar levels get high, the pancreas emits the hormone insulin. The insulin tells the body to remove sugars from the bloodstream. When blood sugar levels get low, the pancreas emits the hormone glucagon. Glucagon signals the liver to break glycogen into glucose and let it back in the bloodstream. However, a diabetic human body cannot do the same. Type 1 diabetes is an autosomal disease with dominant or recessive inheritance. Dominant inheritance is if you get the abnormal gene from only one parent, you get the disease. Often, one of the parents may also have the disease. There is a 50% chance of having a child with this condition. It is true for each childbirth. Children who do not inherit the abnormal gene will not develop or pass on the disease. Recessive inheritance is if there are 2 copies of abnormal genes must be present for the disease to develop. Type 2 diabetes does have genetic components, but it merely increases its risk. Environmental factors play a major role in type 2 diabetes. There are multiple chromosomes and locations on them in which mutations can occur to cause diabetes. Specifically, type 1 diabetes is a mutation of the gene IDDM, which stands for insulin-dependent diabetes mellitus. There is no known cause, but the risk of developing type 1 is increased by certain variants of the HLA-DQA1, the HLA-DQB1, and the HLA-DRB1 genes. These genes belong to a family of genes called the human leukocyte antigen HLA complex. In people with type 1 diabetes, the immune system damages the insulin-producing beta cells in the pancreas. Damage to these cells impairs insulin production and leads to the signs and symptoms of type 1 diabetes. There are not many symptoms, however, when symptoms do occur, they include excessive thirst or urination, fatigue, weight loss, or blurred vision or itchy skin. Since the cells can't take in the glucose, it builds up in your blood. High levels of blood glucose can damage the tiny blood vessels in your kidneys, heart, eyes, or nervous system. That's why diabetes can eventually cause heart disease from high blood pressure, stroke, kidney disease, blindness, and nerve damage to nerves in the feet. Aside from type 1 and type 2 diabetes, there is also gestational diabetes. It is often diagnosed in middle or late pregnancy. Because high blood sugar levels in a mother are circulated throughout the placenta to the baby, gestational diabetes must be controlled to protect the baby's growth and development. With gestational diabetes, risks to the unborn baby are even greater than risks to the mother. Risks to the baby include abnormal weight gain before birth, breathing problems at birth, and higher obesity, and diabetes risk later in life. And this is our works cited.
Thanks for watching.